Hello everyone, welcome to our channel. When most people hear the word hantavirus, they think of a rare disease spread by rodents, typically through contact with mouse droppings, urine, or saliva. But a recent outbreak linked to a cruise ship has brought renewed global attention to a rare and unusual exception, the Andes hantavirus, or ANDV, the only known hantavirus capable of spreading directly from one person to another. The concern was heightened after a deadly outbreak aboard the cruise ship MV Hantavirus. According to the World Health Organization, 8 confirmed cases of hantavirus infection were linked to the vessel, including 3 deaths. The ship carried around 150 passengers and crew. Its journey began in Ushuaia, Argentina, with later stops at Tristan da Cunha and St. Helena. Some passengers, including several Americans, later returned home to states including Texas, Georgia, Arizona, Virginia, and California. Fortunately, none have developed symptoms or tested positive so far. Still, the outbreak has triggered close international monitoring because investigators believe the infections were caused by the Andes strain, a rare hantavirus found in South America and known for one highly unusual feature: person-to-person transmission. That unusual ability has raised an important question for scientists, public health officials, and the public alike. Why can Andes hantavirus spread between humans when nearly all other hantaviruses cannot? Researchers have spent years trying to understand what makes this strain different. The first major clue lies in where the virus replicates inside the body. Most hantaviruses infect people, but they do not effectively establish themselves in tissues that would allow easy transmission. Andes virus behaves differently. Studies show that it actively replicates in the alveolar epithelial cells and alveolar macrophages deep in the lungs. These are cells involved in breathing and immune defense. Why does that matter? Because when the virus multiplies there, viral particles can be released directly into the alveolar lumen, the tiny air spaces where oxygen exchange happens. In simple terms, the lungs become a source of virus particles that may be coughed out. Even more importantly, infected immune cells, especially macrophages, can move upward from the deep lungs into the larger airways. That means infectious particles can potentially be expelled through coughing, heavy breathing, or close face-to-face contact. That gives Andes virus a pathway most other hantaviruses simply do not have. But the lungs are only part of the story. Researchers also discovered that Andes virus replicates in the submandibular salivary glands, the glands under the jaw that produce saliva. Viral material has been found in the secretory cells of these glands and even in the ducts that release saliva into the mouth. That means infected saliva may also carry the virus. This creates a second route of exit from the body, through saliva, making activities like kissing, sharing utensils, or other close personal contact potential opportunities for transmission. This dual shedding, from both the respiratory tract and saliva, is one of the key biological reasons Andes virus stands apart. The next important factor is infectious, viral shedding in body fluids. Unlike other hantavirus strains, Andes virus has been detected not only in blood, but also in saliva, nasopharyngeal secretions, and gingival crevicular fluid, which is fluid found around the gums. In one major prospective study involving 131 confirmed cases, researchers found that during the acute phase of illness, up to 16 days after symptoms began, viral RNA was present in 30% of gum fluid samples, 12% of saliva samples, and multiple respiratory samples. Even more striking, nearly half of those positive fluid samples contained live, infectious virus when tested in cell culture. In other words, this wasn't just harmless viral debris, it was active virus capable of infection. And the timing matters too. Andes virus can be present in the bloodstream up to 2 weeks before symptoms begin, and it remains there throughout the illness. This prolonged viremia, or virus in the blood, gives it more time to spread, especially during the early phase, when people may not yet realize they are sick. This is the early stage of illness, when symptoms look very much like the flu: fever, body aches, fatigue, headache, and sometimes stomach upset. During this stage, viral load is often at its highest. A person may feel mildly ill, but still attend family gatherings, travel, go to work, or care for loved ones. That creates opportunities for close contact and possible transmission. Scientists have also identified molecular features that may help explain why ANDV spreads more efficiently. At the center is the virus's nucleocapsid protein, often called the N protein. This protein in ANDV contains a unique phosphorylated serine at position 386, along with a highly variable region that allows it to strongly suppress interferon beta, or IFN beta, a key part of the body's early antiviral immune response. In plain language, ANDV appears unusually good at hiding from the immune system. That gives it time to multiply to higher levels before the body can react. Higher viral load means more virus in tissues, more virus in secretions, and potentially more opportunity for spread. Genetic studies have also identified specific mutations in its glycoproteins and non-structural proteins, which may improve its ability to infect human cells and move between people. But biology alone does not explain everything. Human behavior matters too. Studies show that ANDV transmission usually requires close or intimate contact. High-risk activities include deep kissing, sexual contact, sharing food or utensils, sleeping in the same bed, and possibly breastfeeding. A major example came during the 2018 to 2019 equine outbreak in Argentina, where 34 people were infected and 11 died. Researchers found that transmission was driven largely by a few so-called superspreaders. These were individuals with very high viral loads, often associated with liver injury, who attended crowded social events while symptomatic. Interestingly, genetic analysis showed very little change in the virus between spreaders and non-spreaders. That suggested the main driver was not viral mutation but social ecology, the pattern of human contact. When health authorities enforced isolation and quarantine, the virus's reproductive number dropped from 2.12 to 0.96, effectively stopping sustained spread. It tells us that while person-to-person transmission is real, it can be controlled using standard infection prevention measures. The ANDV can spread between people, but it is not highly contagious like COVID-19 or measles. According to the Centers for Disease Control and Prevention, the current risk to the American public from this outbreak remains extremely low. And, importantly, the rodents that naturally carry ANDV are found in South America, not in the United States. That means the primary source of infection here remains extremely uncommon. For most hantaviruses, including those found in North America, the main risk is still rodent exposure. That means avoiding aerosolized rodent droppings is critical. If you need to clean an area contaminated by mice, experts recommend spraying the area thoroughly with a bleach solution, 1 part bleach to 9 parts water, and letting it sit for at least 5 minutes before wiping it up with paper towels. Do not vacuum or sweep dry droppings that can launch viral particles into the air. Finally, always remember to wash your hands thoroughly after cleanup, and a mask can offer added protection. That's all for today. If you found this content interesting and helpful, don't forget to give a thumbs up, subscribe, and hit the notification bell. Thanks for watching, and we'll see you next time.